Hi, my name is Jasmine Prieto, and for my fourth sports medicine project, I am doing medical journal research for genetic editing. What is genetic editing? Genetic editing uses CRISPR-Cas9, a technology that allows scientists to modify an organism's DNA. It can be used in medicine to treat genetic disorders, in agriculture to improve crop resilience, in animal breeding for desirable traits, in biotechnology for sustainable materials, in research and gene therapy to understand/treat diseases, in synthetic biology for new biological systems, and in conservation to help endangered species. The research I did mostly focused on medicine to treat genetic disorders and synthetic biology. How does it work? Genetic editing works through a few key steps, often using the technology CRISPR-Cas9. The first step is targeting the DNA, which involves identifying the specific sequence of DNA that needs to be edited. Scientists then design a short RNA sequence that is complementary to the target DNA. This RNA is known as a guide RNA, also known as gRNA. The second step is cutting the DNA. The guide RNA is combined with the Cas9 protein, which acts as molecular scissors. When introduced into the cell, the gRNA binds to target DNA sequence, and Cas9 cuts the DNA at the precise location. The next step is repairing the DNA. After the DNA is cut, the cell's natural repair mechanisms kick in. There are two pathways for repair: the non-homologous end joining and the homology-directed repair. After that comes the resulting changes. The outcome can vary depending on the method used. For example, genes can be knocked out to study their function, or new genes can be inserted to give the organism new traits. The final step is applications by correcting the underlying genetic issues for disorders.
CRISPR-Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then, they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA-cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing.
The two types of repair methods from step 3 are the non-homologous end joining, also known as NHEJ, and the homology-directed repair, known as HDR. The NHEJ method often introduces small insertions or deletions at the cut site, which can disrupt the gene and knock it out. This method is also the most commonly used method in genetic editing because it is quicker and more straightforward. The HDR method is if a donor DNA template is provided. The cell can use this template to repair the cut, allowing for the insertion of new genetic material or precise modifications, which makes this method more complex and time-consuming. Targeting genetic disorders. CRISPR-Cas9 targets genetic disorders by using the process of step 2, which is a specific guide RNA to locate the faulty gene in the DNA. Then for Cas9 to successfully bind and cut the DNA, a specific sequence called the protospacer adjacent motif, also known as PAM, must be present next to the target sequence. This PAM sequence helps the Cas9 identify the correct location in the genome. This approach has shown promise in clinical trials and holds the potential to provide a long-lasting cure for sickle cell disease in the near future. The PAM sequence required for the Cas9 protein is typically NGG, where N can be any nucleotide between A, T, C, or G. What is sickle cell disease? Sickle cell disease is a genetic blood disorder caused by a mutation in the HBB gene that affects hemoglobin, the protein in red blood cells responsible for carrying oxygen. In sickle cell disease, the red blood cells become rigid and shaped like a sickle instead of the normal round shape. This abnormal shape can lead to blockages in blood vessels, causing pain, anemia, and increased risk of infections. The disease is inherited in an autosomal recessive pattern, meaning a person must receive 2 copies of the mutated gene, one from each parent, to develop the disease. Treatment options focusing on managing symptoms and preventing complications, but advances in genetic editing like CRISPR are being explored as potential cures. Curing sickle cell disease. The goal of curing sickle cell anemia starts with the specific guide RNA sequence for targeting the mutated HBB gene in sickle cell disease. Can vary on the exact mutation and the design of the CRISPR-Cas9 system. However, one commonly targeted mutation for sickle cell disease is the one single nucleotide change from A to T in the 6th codon of the HBB gene. For example, a guide RNA sequence designed to target the sickle cell mutation might look something like this. The guide RNA sequence. This sequence would be complementary to the DNA sequence near the mutation site in the HBB gene, guiding the Cas9 protein to the precise location for cutting. The adjacent PAM sequence would be necessary for the Cas9 protein to recognize and bind to the target DNA. Given this guide RNA sequence, a possible PAM sequence immediately following the target DNA sequence would be GGG. By using this guide RNA, the CRISPR-Cas9 system can specifically target and edit mutated HBB gene, providing a potential cure for sickle cell disease. Designer babies. The concept of designer babies involves using genetic editing to select or modify traits in embryos. This could potentially allow for the selection of physical attributes, intelligence, or disease resistance. However, it raises significant ethical, social, and safety concerns and is highly controversial. How it works. Genetic editing for designer babies would work basically the same as targeting genetic disorders using CRISPR-Cas9, but to modify the DNA of embryos instead. Scientists could potentially add, remove, or alter specific genes to achieve desired traits. This process would typically involve gene identification, which is identifying the genes responsible for the desired traits, then editing process, using CRISPR-Cas9 to cut the DNA at specific locations and introduce changes, then embryo development, allowing the edited embryos to develop, ensuring the changes are successfully incorporated, then screening the embryos to confirm the desired genetic modifications. Benefits. The potential benefits of designer babies could include disease prevention, reducing or eliminating genetic disorders, or enhanced traits with possibility of selecting traits like intelligence, physical abilities, and appearance. Longer lifespan, potential to enhance genes related to longevity and health, or improved quality of life, overall better health, and fewer medical issues. Risks. Some potential risks of designer babies include unintended consequences, which are unknown long-term effects and possible new health issues, ethical concerns, which are moral debates about altering human genetics, then social inequality, widening the gap between those who can afford genetic modifications and those who can't. Lastly, genetic diversity, which is potential reduction in genetic diversity, which could affect the population's resilience to diseases and environmental changes. Challenges. When genetic editing in humans becomes available, the biggest challenge for all uses will be that it is about $2.2 million per treatment. Along with that, some challenges for designer babies include ethical dilemmas, regulatory issues, technical limitations, public acceptance, whereas some challenges of genetic editing diseases include delivery methods, off-target effects, regulatory approval, and ethical considerations. Expectancy. Genetic editing in humans, particularly through methods like CRISPR, holds great promise for preventing genetic disorders, enabling personalized medicine, and enhancing traits. However, it also raises significant ethical and regulatory challenges that need careful consideration. Genetic editing in humans is expected to see advancements in several areas. When we may start seeing more widespread applications in the next decade, particularly in treating genetic disorders, cancers, and certain hereditary diseases. Clinical trials are already underway for conditions like sickle cell disease and muscular dystrophy, where genetic editing is likely to be implemented in specialized medical centers and research institutions around the world. Countries with advanced biotechnology sectors such as the United States, China, and various European nations are leading the way in research and development. Overall, the future of genetic editing in humans holds promise for treating and potentially curing various genetic conditions. Conclusion: genetic editing in humans offers remarkable potential to eliminate hereditary diseases and enhance health. However, it raises critical ethical concerns regarding natural evolution and the long-term impacts on future generations. Balancing scientific advancement with ethical responsibility is essential to ensure that genetic editing benefits society without compromising fundamental human values. Thank you.